Olabanaki everyone, welcome to this video about polymers. This is where chemistry starts to get actually useful and that we can relate it directly to things that we see in everyday life. So different types of polymers including DNA, proteins, all the plastics that we use in everyday life, and some other polymers such as nylon, Kevlar, terylene, etc. In level 2 we're going to look at addition polymers, and in level 3 we need to know about condensation polymers. Let's start with addition. In general, polymerisation is where we take millions of tiny units called monomers and we join them together in long chains called polymers. To make a polymer, we need quite a few conditions, usually heat, pressure, and certain catalysts. You don't need to know the specifics of each different addition polymerisation reaction. The general mechanism of addition polymerisation starts with a carbon-carbon double bond. So each of these little molecules above, each ethene is a single monomer. When we apply heat and pressure, the double bond breaks and the freed up electrons are used to join to adjacent monomers, and this creates an incredibly long chain. The resulting polymer is joined by many carbon-to-carbon single bonds, and since these are strong and resistant to reactions like hydrolysis with water, the resulting plastics they make are very long-lasting. They are very resistant to breaking down, and so an environmental problem exists, one of which I'm sure you're aware of. Let's compare addition and condensation polymerisation. The diagram at the top is addition polymerisation. All monomers start with a carbon-to-carbon double bond, which is broken under conditions of high pressure, heat, catalysts, and single carbon-to-carbon bonds are made to form the addition polymer chain. Whereas condensation polymers employ a condensation reaction. This is a reaction you should have studied in level 3 chemistry already, and these are our level 3 polymers. There are two types of condensation polymers as shown here. Polyamides, where you make an amide bond to link the monomers of the chain together, and polyesters, where you make an ester bond to link the individual monomers together. Let's take a closer look at the polyesters. There's two ways to make all condensation polymers. We're going to look at one where we use two different monomers. As you can see here, the first monomer has two carboxylic acid groups, and the second monomer has two alcohol groups. So if we remove the OH from the carboxylic acid and the hydrogen from the alcohol and form a bond in there, we form the ester group. So we remove 1 mole of water for every ester bond we make. Note also that we have the familiar ... on each end of the polymer showing that this does not terminate, or it goes on and on and on for quite a while. Now, the example we just looked at was using two different monomers. Let's have a look at how we can make a condensation polymer from a single monomer. Let's start in the reverse direction this time. Let's start with the polymer. This line here is the start of the polymer, and we need to go along to the right until we start to repeat again. The part between the lines is called our repeating unit, or our monomer. And what we need to do is we need to copy that out and we need to remake the original functional groups. You can see in here we've added an H in to that left-hand O to make an alcohol group, and we've added in an OH to the C double bond to O to make a carboxylic acid. You can see here that the monomer also has two functional groups, but this time they are two different functional groups, unlike the last example. Two different functional groups that can be used to make an ester. So an alcohol and a carboxylic acid, or an acyl chloride and a carboxylic acid. Let's now take a look at a very special ester, fats and oils, which are triesters of 3 fatty acids and propan 1,2,3 triol, otherwise known as glycerol. Looking at the reaction here, we have glycerol on the left plus our 3 fatty acids. So if we take the H from this alcohol group and the OH from one of the carboxylic acids on the fatty acid and remove it as water and form our normal bond from condensation, we'll make an ester group. If we repeat this 3 times, we get 3 ester bonds, one for each of the fatty acids onto each of the OHs on the 1,2,3 propan triol. The next thing to notice is what these R groups stand for, and this is what makes the acids fatty. They are long carbon chains. They can have double bonds in them, and as you can see by the single dash, the double dash, and the triple dash, they can all be different. And finally, just a good point to note is that the carbon-to-carbon single bonds from the propane triol do not break. So you actually end up with one very large molecule, our fat or our oil. Turning our attention now to polyamides. In the same way as polyester, we're going to remove a small molecule in a condensation reaction. If we start with a dicarboxylic acid as one monomer and a diamine as the other, we'll remove the OH and one hydrogen from the amine as water to form the new bond, the amide bond. So remember that one water is removed for every bond formed over and over again in this polymer. And we can also use a diacyl chloride instead of a dicarboxylic acid, in which case the small molecule removed would be HCl instead of H2O. Our first special example of polyamides are actually proteins and peptides. A peptide is just a small protein. The monomers for proteins are called amino acids. You can see a couple of examples here, alanine and glycine. Each amino acid has a central carbon atom bonded to a hydrogen, an amine group, NH2, a carboxyl group, COOH, and a side chain. These are varied, and the side chains gives the amino acid its identity. Note that only glycine has a side chain of hydrogen, and so it is the only one that cannot form optical isomers, as it doesn't have 4 different groups attached to a chiral carbon. If we now look at how proteins are made, you can see in this circle in the blue, removing the H and the OH again in a normal condensation reaction to form an amide bond in purple. But the difference is this time that proteins and peptides do terminate. You can see the left-hand side has an NH2 and the right-hand side has a COOH. So there's no dots in proteins and peptides. Note also that you can have as small as a dipeptide, which is simply 2 amino acids joined by an amide bond. A tripeptide is 3, and a protein has many, a few hundred at times. Our next polymer of interest is Kevlar, commonly used in bulletproof vests and other very strong pieces of armour. The 2 monomers used to make Kevlar are shown here. The diamine on the top left, and this time a diacyl chloride, which means for every amide link made, we remove HCl instead of H2O. Another important and common polyamide is nylon. This version of nylon is called nylon 6,6, and the 6,6 is to do with how many carbon atoms there are in each monomer, as shown here. If we now take a line through the middle of the amide group and split this back up, you'll see the original monomers were a diamine and a dicarboxylic acid. However, what's drawn here is a single repeating unit consisting of one of each of the monomers in this case. And here you can see some of the uses of nylon 6,6. And that brings us to the end of this video. Polymers can be quite confusing and there's many different types of them. So make sure you focus on the condensation part and the 2 functional groups of amide and ester. The rest is simply copying. In the next video, we're going to look at the hydrolysis of the condensation polymers. See you then.